Hi, I'm Will from BiologyWise, and in this video, we're going to be looking at B lymphocytes and humoral immunity. Like other lymphocytes, such as T lymphocytes, B lymphocytes are a type of lymphocyte that are able to detect specific pathogen antigens, allowing them to produce a specific response to specific pathogens. The first stage of the B cell response involves the detection of antigens. B lymphocytes, or B cells, detect these antigens via antibodies on their cell surface membrane that act as antigen receptors. These antibody receptors are unique to each individual B cell, with each having a different three-dimensional shape. This means that each B cell can only detect an antigen if it matches the shape of its specific receptor. If the receptor and antigen match, they will bind, causing the receptor and the antigen to enter the B cell via endocytosis. The antigen is then processed and presented on the surface of the B cell. The next stage of the B cell response involves B cell activation. B cells cannot activate on their own after detecting an antigen. Instead, they must be activated by a T helper cell. The T helper cells are able to activate other immune cells, such as B cells and phagocytes, and are produced during the T lymphocyte response. The T helper cell is able to bind to the antigen once it has been displayed on the surface of the B cell. This binding activates the B cell, stimulating it to rapidly divide via mitosis, producing genetically identical clones in a process called clonal selection. The B cell clones produced during clonal selection can develop into one of two types of specialized B cell. These types are called plasma cells and memory cells. Plasma cells are responsible for the production of antibodies, being able to produce several thousand antibodies every second. Memory cells circulate in the body for many years and can recognize the pathogen responsible for the original infection. If the memory cell detects this pathogen in the future, it can rapidly divide into new memory cells as well as new plasma cells that produce antibodies that can target the specific pathogen.